let's skip over that. Number 5, somatic hybridisation. What is somatic hybridisation? I think you're going to get a copy of this paper also in your handout. If you're particularly interested in what I'm about to talk about, you can go back and read this. Basically, somatic hybridisation is an asexual process by which rust isolates can mix up their genetic material. So it's like sexual recombination, but without the sexual recombination, if you like. Now I'll explain what I mean by that, okay? The first thing that's very important to understand somatic hybridisation is that individual rust isolates on wheat are dikaryotic. Do you know what I mean by this? 2 nuclei per cell, exactly. That's what this diagram means. This is, let's just call this pathotype 1. Each cell has 2 nuclei. So it's a diploid organism, but it has 2 haploid nuclei. All right, it's a functional diploid, but it has 2 haploid nuclei. And what we've found with rust isolates, these rust isolates, is that these 2 nuclei are genetically, invariably, genetically very different. So you don't have 2 nuclei in a rust that are identical. There's usually a huge degree of genetic difference between the 2 nuclei. And that's what makes them so difficult to work with genetically. We're now doing genome sequencing with rust isolates. It's really hard because whenever you sequence one genotype, you're almost trying to sequence 2 genomes that are mixed up together. It's really hard to separate them out and do all the genome annotation and that work because of this dikaryotic nature. All right, so just think of that as pathotype number 1 with those 2 nuclei. Here we have another pathotype which also has the 2 nuclei that are different, but they're different to the ones that occur in pathotype number 1. What we believe happens, and Zach showed some pictures of this before, you can actually get hyphae or germ tubes of rust that grow across each other, and we certainly know this happens with other fungi, and they fuse, they join, produce like a bridge, and what we can get is the mixing of nuclei across that bridge. All right? So this diagram is a very simplistic example where we have one pathotype that has fused with another one through an asexual process, and then nuclear exchange has occurred, so we end up with a hybrid that combines this nucleus from this isolate with that nucleus from that isolate, and here's the reciprocal one. All right? So these are 2 hybrid isolates that are potentially different pathotypes that have been generated through what we call asexual or somatic hybridisation. It's a very, very rare process, but we do know from our studies in Australia that it does occur. All right, so we have at least 3 examples of this process from our long-term studies in Australia. First one, interestingly, is between 2 former species of Puccinia graminis. So we have a grass in Australia that's infected by Puccinia graminis tritici, the wheat stem rust pathogen, and it's also a common host for the rye stem rust pathogen, Puccinia graminis former species cicalis. And we have hybrid isolates that we get off that grass that are intermediate between the 2. They can infect barley, so we get a 3rd form of stem rust off barley as well, this hybrid. They can't infect wheat, they're highly avirulent on wheat. They can infect rye, they can infect barley. We believe these are asexual hybrids caused by this exchange of genetic material between the wheat stem rust pathogen and the rye stem rust pathogen. We also have an example from the 1950s where we had 2 pathotypes of the wheat stem rust pathogen undergo somatic hybridisation to give a new pathotype altogether. Right? And this actually resulted in at least 2 varieties becoming susceptible. So it actually it was important in agriculture, this hybrid rust. And then the last one was one that I found in the 1990s where I found a pathotype, don't worry about these numbers here, but a pathotype of the wheat leaf rust pathogen, Puccinia triticina, that was clearly a hybrid between 2 of our pathotypes that were quite common at the time. And again, this hybrid leaf rust pathotype was quite important in rendering a number of wheat varieties susceptible. The really interesting part about that story is that we had a hybrid rust, this one down here, and it was actually virulent on hybrid wheat. We had a hybrid wheat breeding program that had 3 or 4 varieties, and this hybrid rust was able to infect the hybrid wheat. Quite an interesting story, but I won't go into that in too much detail now. If we look at this scabrum rust, okay, so this is the one where we've got...
Just these races or phenotypes, are they all in the collection? Do we have them in the
Absolutely, we do.
I think it would be interesting to check their molecular weight if you have done.
Well, so this one here, the scabrum rust,
Yeah.
that's some SSR fingerprints, all right? So what we have here, this is a student of mine, Lida Derevnina. I got her to do this project. So we took 14 isolates of this scabrum stem rust, which we believe is a hybrid between wheat stem rust and rye stem rust. We extracted DNA and we did fingerprinting on them. So here's the 14 isolates of scabrum rust. These are wheat stem rust, Puccinia graminis tritici, a couple of Puccinia graminis cicalis. This is the oat stem rust pathogen and the rye grass stem rust pathogen. But first thing to notice from this is that there are different genotypes within those hybrid isolates. So the question that we've often asked is, okay, we believe we have this somatic hybridisation going on, but how often does it happen, and has it happened more than once? Well, these results tend to suggest, right, that we've had multiple hybridisational events, because each of these, quite a few of these isolates are actually different. This one's different to that one. This one, I guess you would say, is the same as that one. This one is different to that one. So it appears as if there have been multiple hybridisational events that have occurred between the wheat stem rust pathogen and the rye stem rust pathogen. That's the first thing to notice. The other thing to notice is that some of the hybrid isolates combine alleles from the parents. So here's the wheat stem rust allele, and there it is there. Here's the rye stem rust allele, and here it is there. So these hybrids here appear to combine the alleles, those 2 SSR alleles, from their parents. Okay, so I won't go into any further detail about that, but it's just an interesting example of what we can do with these historical isolates. We can now go back and see, like you said, Sajid, look at them and try and figure out, you know, how many times this hybridisational event has occurred, and also whether or not there's been any further change in virulence, and we've got evidence for that, where some of these hybrids we believe have then subsequently mutated and acquired virulence for resistance genes in wheat. We do some of this work in the greenhouse, so we try to, some of our students have tried to induce hybridisation in the greenhouse, and the way we've done this is to use colour mutants. So we can use spore colour as a marker to show us whether we've had or evidence of hybridisation. So what we've got here is an albino isolate of wheat stripe rust. It's white. It's a mutant we picked up in the greenhouse that just spontaneously lost its ability to produce pigment in its spores. So it's an albino mutant, and here's a normal coloured or a wild type colour variant of pathotype of wheat stripe rust. What we've done is we can mix them together and inoculate them onto a common host and get them to grow together, right? So if you look at that close up, we can get the colonies to grow into each other, and hopefully we'll get hybridisation occurring, all right? So we've done these experiments, and we've actually picked up some isolates that are intermediate in their colour. So here's the albino, here's the wild type, and this one looks intermediate in its colour. We suspect this could be a hybrid. So we're now doing some DNA fingerprinting on this to try and figure out whether it actually is a result of a hybrid, or it could be a mutant in the greenhouse that's just acquired. It could be a mutation of this one that's just acquired, reverted back to the ability to produce yellow pigment in the spores. And we have examples of this as well. All right, so we do believe that somatic hybridisation is going on in nature. We're not entirely sure what the mechanisms are. Simplistically, we think it's nuclear exchange, but we're now doing some studies under controlled conditions to get a better understanding of that. All right, so that's pretty much it. Just to recap, 5 different mechanisms we believe that generate genetic and hence pathogenic variability or different pathotypes of the rusts. Mutation we believe is the most common, and the frequencies of the others not so common. One final comment to make, and we're going to be talking more about identifying pathotypes and things like that. A lot of our work in the past, or up till now, is focused on pathogenic change with respect to major gene resistance. So I showed you that picture before with the mutation to virulence on YRA, YR6, YR7. These are major genes, they're seedling genes. We're now using a lot of adult plant or minor resistance genes. We haven't really looked at, critically, whether or not these pathogens can acquire virulence on these minor genes. And there is some evidence, well, we've shown at least 1 or 2 cases where they have. If we're going to use these adult plant resistances, these minor gene resistances more and more commonly in future, then it makes sense that our work on the variability being generated in these rust pathogens should also look at changes, potential changes with respect to virulence on those minor genes. The other one down the bottom, pathogen aggressiveness, we haven't mentioned this yet, but we do know that some isolates, some pathotypes grow quicker, produce more spores, can grow better at higher temperatures. We know this. Sajid has done a lot of work on this, I think, in his PhD. Or you've actually looked at pathogen aggressiveness in Denmark with Mogens, I think? Yeah.
In beast, I've done that.
Okay, so there are people now looking at this too. This is also potentially an important trait. Another, let's say another genetic variation in the pathogen genome that can give us potentially changes or increased susceptibility in an agricultural context.